Note taker 12, Continuing Genetics. On the last note taker, we talked about Gregor Mendel and his pea plants, and we learned that each parent plant had a pair of factors, or a gene pair, for each trait. And through the process of meiosis, each parent can only give one gene from the pair for each trait. So when we pass those genes down to offspring, we have what are called inherited traits. So inherited traits are characteristics that are passed from the parent to the offspring. Now, when we talk about inheriting traits, there are two laws that were formed that follow along with the laws of genetics. The first is the law of segregation. And if you can remember that to segregate means to separate, you'll understand this law. And you'll also notice that it has the word separate in the definition. The law of segregation states that each of the parent's gene pairs, so the gene pair that they have for hair color, the gene pair that they have for eye color, skin color, whether they have freckles, whether they can curl their tongue or not, each of these pairs must separate during the process of meiosis, and one gene will go to each new cell that's being formed. So if the mom has a dominant trait for freckles and a recessive trait for no freckles, when her body is forming egg cells through the process of meiosis, one of the egg cells will get her dominant trait, one of the egg cells that's made will get the recessive no freckle trait. That's the law of segregation. The second law is the law of independent assortment. It means that all of your characteristics you have are inherited independently of each other. We don't have many traits that combine together. We're going to talk about one later in this note taker that does, but to start off, while we're working on this one, we're going to be under the assumption that let's talk about hair color and eye color. Just because my hair is brown doesn't mean I have to have blue eyes. I can have brown hair and green eyes because my hair color pair and my eye color pair are inherited independently of each other. They're not connected to each other. In addition to the laws of segregation and the law of independent assortment, we're going to talk a little bit briefly about a situation called incomplete dominance. Mendel showed us that some traits are strong, so we call them dominant, and some traits are weak, so we call them recessive. And for the most part, your traits work that way. However, we do have some situations, even in us, like our skin color, that if you look around the room, there aren't just two skin colors in the classroom. You have lots of skin colors. So what that led scientists to do was study these traits, and they realized that it's not just one dominant and one recessive that makes this up. We can actually have lots of different alleles that code for the same type of trait. Now, when we have two dominant traits that blend together, we have a situation called codominance or incomplete dominance. I'm going to talk about a couple of situations on the next few slides, and I will show you pictures to go with them. The scientist who first discovered incomplete dominance was a German botanist by the name of Karl Corns. And what he did, he worked with these pretty little flowers called 4 o'clock flowers. And he showed that there are 3 color variations that they can be. They can be red, they can be white, or they can be pink. Now, at the beginning, he assumed that the red flowers were homozygous dominant. So that would mean that the red flowers were capital R, capital R. And he assumed that the white flowers were recessive. which should have meant that they were little r, little r. Well, when you cross a red flower and a white flower, the red flower would give a capital R, the white flower would give a lowercase r, and since the pair had a dominant, they should have had red flowers as offspring, but they didn't. When he crossed them, they ended up with pink flowers. So he had to go back and rethink what was going on. And what he determined was the red flower was definitely homozygous dominant, capital R, capital R. And the white flower was also homozygous dominant, capital W, capital W. Now when you cross them, you end up with a gene pair that has a dominant red and a dominant white, so you have the gene pair RW, and you get a blending of the colors to give us pink. Here is a picture of those flowers. On the top left is our red flower, on the top right is the white flower. So the red flower can only give an R to its babies. The white flower can only pass down a W to its babies. So when you join them together, you get the gene pair RW, which they're both dominant. So the red and the white, they blend together and you get a pink flower as their offspring. Incomplete dominance doesn't just occur in plants. It can also occur in animals. And I have here the example of horses. If you look at the top left horse, that is a pure chestnut or brown horse. Its gene pair is capital B, both dominant to represent the brown color. And on the top right, we have a pure white horse, which is dominant W to represent the white color. The brown horse can only pass a dominant brown color to its baby, so it has the capital B. The white horse can only pass a capital W. And when you get that blending between the brown and the white, both being dominant, you get the horse at the bottom, which is a palomino, which is a light tan color with a blonde mane and a blonde tail. Incomplete dominance also works with our skin colors. There are about 8 different alleles, so we use 8 different letters to represent our skin color genes. And depending on which coding or which pair you get will determine what skin color you have. In the pictures on the previous slides, you saw that I used arrows to just show that the brown horse had given the offspring, the baby horse, the B, and the white horse had given it a W. But there is actually a better way to make predictions about what an offspring is going to look like if you know what the parents have. We're going to be working with special charts called Punnett squares to actually show possible gene combinations and make predictions about what traits the babies are going to have. Now, before we can start with the Punnett squares, there are 2 vocabulary words that you have to know. And these are 2 that you're going to have to memorize, you're going to have to know the definition of, because we're going to be working a lot over the next few classes with Punnett squares, and you're going to see the words phenotype and genotype quite often. And when we take quizzes on these, you are going to have to know how to count percentages, but you're going to have to know what we're talking about using these words. A phenotype is what you physically look like. So if you look at the PH at the beginning of the word phenotype, that's what you physically look like, what you can see. So I can see somebody has brown hair. I can see somebody has blue eyes. Or I can see somebody has freckles. That's a phenotype. Your genotype is the actual gene pair of letters that make up your characteristic. So my phenotype is I have freckles. You can see them. That's my phenotype. My genotype is a capital F and a lowercase f. And I'll talk to you later on about in class how I know that that's what I am. So in the Punnett squares, we will need the genotypes the gene pairs for the parents to figure out what the genotypes or gene pairs of the offspring are going to be. But we're also going to count percentages for phenotype, so you are going to have to know what these genotypes, these gene pairs represent. On this slide, I'm going to talk briefly about how to set up a Punnett square, and then on the next slide, I'm going to actually have one completed for you. If we're doing combinations for a single trait, which is all we're going to look at and concentrate on here in 7th grade, you need to draw a box that has 2 columns and 2 rows. After that, you're going to need to know what is mom's genotype, her gene pair, and you're going to need to know what is dad's genotype or gene pair. Now, I will tell you the best advice right here. When you are setting up a Punnett square, always set it up the same way. I've been doing Punnett squares since I was your age, and I always set it up the same way. I put the mom's genotype across the top, and I put the dad's genotype down the side. Now, I don't care if you want to put dad's pair on the top and mom's down the side, but however you do that, do it the same every single time so you get into a pattern, and then Punnett squares will become much easier for you. Here in this Punnett square, I have a cross between a mom who is heterozygous, capital B and lowercase b, and a dad who is also heterozygous for a trait, capital B, lowercase b. And let's say that this represents freckles. So freckles are dominant, so mom has freckles because she has a capital B. Dad has freckles since he has a capital B in his pair. Now, how to fill in the boxes. The boxes that are white in this Punnett square represent what the possible offspring can be. So the boxes represent what pairs the babies are going to have or can have. I have color-coded mom's pair and dad's pair so you can see where the letters are going each time. So mom has a capital B and a lowercase b. So her capital B goes over one column. Mom's lowercase b goes over the other column. Dad I represented with blue and yellow. Dad's capital B goes in front of the first row. Dad's lowercase b goes in front of the second row. Now, we're just going to combine these letters as they join in the boxes. So if you look at mom's capital B, that capital B drops down into each box below it. So that's why there's a red B in both boxes underneath of mom's red capital B. You'll notice in mom's green lowercase b, in all the boxes underneath, you have the green lowercase b. Dad's big B actually goes across since his is in front of a row. So you'll notice that each box by the blue capital B, each one has a blue capital B. And then the yellow B goes into each box beside it. Now, I could have written, you'll see that arrow there, I could have written lowercase b, capital B. It's the same exact thing as capital B, lowercase b. It's just proper that the capital letter comes first when we're writing it. Don't worry if this is hard to understand. We're going to be doing plenty of Punnett squares in class for you to practice with. Once we have completed the Punnett square, now we do the important part, and we try to see what the probability or likelihood that the offspring will have a certain trait. And we're going to show this probability in percentages. So all 4 boxes have to equal 100%. And since there are 4 boxes, each one is going to be worth 25%. So if you think of it as 4 quarters making up $1. So each box is worth 1/4. Now, if I have none of the boxes, if what I'm looking for is not in any of the boxes, then I'm going to have 0% chance. If what I'm looking for is in one of the boxes, then I have a 25% chance. If what I'm looking for is in 2 boxes, it would be 50%, 3 boxes, 75%, and 4 boxes would be 100%. I mentioned earlier that most of our traits are inherited independently of each other. However, in the early 1900s, there was a zoologist, somebody who studies animals, named Thomas Hunt Morgan, and he started working with fruit flies. And fruit flies don't have many chromosomes at all, so they're super easy for scientists to work with. And what he noticed was that at a certain pair, when he looked at them, all the females had the same looking pair at one of them, but the males had 2 different chromosomes there. So if you look at the picture on this slide, that would be what he saw under the microscope for all the males. All the females, if you look at the second, the larger chromosome, all the females had 2 of those. And because of the way they're shaped, he called them the X and the Y chromosomes. And he realized that these chromosomes must have something to do with determining the gender of a living thing. Here are what we call karyotypes. And you'll notice that 22 of our 23 pairs are numbered, but you'll notice that the last pair, you have an X and a Y. So if you look at the karyotype on the left, this represents a male. If you look, he does have one shorter Y chromosome and one of the larger X chromosomes. But if you look at the karyotype of a female, you'll notice that she has 2 larger chromosomes at the X and none at the Y. This led scientists to conclude that the X and Y determine the sex or gender of an offspring. If you are a female, you have 2 X's. So your gene pair that makes you a female is XX. If you are a male, your gene pair that makes you a male is XY. Now, as scientists continued to study this, they found that genetics is so not as easy as it could be, that they keep finding new stuff out. And they found that not only do we have chromosomes that determine what gender we are, but there are actually some characteristics or traits that attach themselves on to the X chromosome. So when we pass the X chromosome down to our children, those traits are linked to that and can also be passed down. These are called sex-linked traits, and they're going to be attached to the X chromosome. We are going to represent those sex-linked traits as little letters that look like exponents. So if you think of the little exponent there, the little H attached onto an X, wherever that X goes, that little H has to follow. So it's almost like you wearing a backpack. Wherever you go, your backpack follows. Your little backpack would represent the sex-linked trait following you around. Now, there are a couple things I want to point out in this Punnett square below. This is a Punnett square that shows the sex-linked trait for hemophilia. Hemophilia we're going to talk about in class, but it is called the bleeder's disease. It's when you do not have, or when your body does not produce enough platelets to stop you from bleeding when you cut yourself. So hemophilia can actually be very serious. People can die from it because they can cut themselves and they can't stop bleeding. Now, Hemophilia is a recessive trait, so to have it, you have to have all little h's. Now, I will also say that because of the way it attaches onto sex-linked traits, it's a lot easier for a boy to be hemophilia, or to have hemophilia, than a girl to have hemophilia. Because a boy only has one X, so if that X has a little h, he has hemophilia. But a girl has two X's, so she has to have a little h on both X's to have the disease. So we have here a Punnett square that shows a mother with hemophilia, so she's X little h, and then we have a healthy father. Since he's healthy, he's X capital H. That capital H represents that he's healthy. And you'll notice that the Y is all by itself because we don't have anything that links to it. Because sex-linked traits only attach to the X, not the Y. And I say that now for 7th grade. When you get to college, you'll find out different. Now, if you look at the Punnett square, I've color-coded these again. Mom's X little h drops down into each box below it. Mom's green X little h drops to each box below it. Dad's X big H goes across into boxes. And Dad's little yellow Y goes across into the boxes. So now if we look at percentages here, there is a 0% chance of having a completely healthy daughter who is not a carrier. She would have to be X big H, X big H. There's no chance of that because we don't have any in boxes. If you want to know what chance they'll have a daughter who is a carrier, it means she's healthy, capital H, but also carries the gene to give it to her children, little h. So we have two boxes that have X big H, X little h, so that would be 50%. We have 0% chance of having a daughter who has hemophilia, because for her to have hemophilia, she would have to have the same pair as her mom and be X little h, and we don't see any of those in the boxes. Now, the boys. We have a 0% chance of having a healthy boy, because he would have to be X big HY, and we don't have any of those. So there's a 50% chance of having a boy who has hemophilia, because he would be X little h, Y. So that's how to read the Punnett square with the percentages. The last thing I'm going to talk about on this note taker is something called a pedigree. A pedigree is a chart or a family tree that's going to track a certain trait through the family. Now, normally we don't do pedigrees for regular traits, like we don't track hair color through a family, or we normally don't track freckles through a family. Usually pedigrees are used to look at problems that might arise, like color blindness or hemophilia. So I'm going to show you an example of a pedigree on the next slide. Don't panic, because we'll be working with more pedigrees in class. Here's an example of a pedigree, and it's in your notes. This is tracking color blindness through a family. Like hemophilia, color blindness is sex-linked and is recessive. So to be colorblind, you have to have both recessive traits if you're a girl, or one recessive trait on your X if you're a boy. So, a couple things I want to point out about this picture. Boys, the males in the family, will always be represented by squares. Females will always be represented by circles. And here, since it's a sex-linked trait, we can have a colored-in circle, an empty circle, or a half-colored-in circle to represent the girls. The boys, you're either going to have a colored-in circle or an empty circle. empty circles and empty squares are going to represent healthy males and healthy females, do not have the disease at all. Squares and circles that are totally colored in are going to be males or females that have color blindness. The circles that only are half colored in are going to be a girl who is healthy. She doesn't have color blindness, but she carries the recessive trait which she could pass to her children. Now, the other things I'm going to show you in here is how to represent when a couple is married or when they have children. If you look at the top square and top circle, you will notice that there is a line passing between them. That means that they are a married couple. To show that a married couple has children, you'll see how there's a line that drops down from the marriage line, and then you see how it's attached to a square, the empty square, the half-filled circle, the empty square, the half-filled circle, and then skip over the next empty square and go to the next one, empty square. That means that the top mom and dad have 1, 2, 3 healthy sons and 2 daughters, the circles with 2 daughters that are carriers for hemophilia. Or sorry, for color blindness, because we were talking about color blindness. However, this chart could also represent hemophilia because it works the same way as color blindness. Again, we will be working more with pedigrees in class. You'll be doing more work and get to make some and read some in class. Please don't forget to answer the 3-question assessment that follows the video.